Hello, and welcome to this Pharma On podcast, part of our DMPK podcast Insights series. And my name's Scott Summerfield. I lead the Metabolism Group, which is part of the UK's integrated metabolism and drug discovery platform. Today, I'm really pleased to be joined by Dr. John Mehl, who's going to discuss protein mass spectrometry for advancing the bioanalysis of biomarkers and biotherapeutics. Welcome, John.
Well, thanks, Scott. Really appreciate the offer to join in on this podcast. I'm looking forward to it.
So, by way of introduction, John has been working in the field of biological mass spectrometry for more than 20 years, currently consulting, but having previously worked with GSK, Merck, and Bristol Myers Squibb. And that's covered support of proteomics, as well as data analysis of small molecules, large molecules, and vaccines. John, perhaps as we start, just describe the kinds of things in protein mass spectrometry that you've been getting involved in recently.
So, our primary remit is the quantification of protein biomarkers and biotherapeutics. But we also get involved in some other applications, namely in vivo characterization or the biotransformation of biotherapeutics. So once these molecules are dosed, what happens to them, right? And another interesting area that we get involved in is target protein turnover. So that's really trying to measure what the in vivo synthesis and degradation kinetics are for proteins that are our targets, our therapeutic targets. And that's really important information when it comes to like how do you do the modeling, how do you predict what dose level you need, how frequent you need to do. So, these are the areas we cover, and it's all built upon using mass spectrometry.
With 20 years, you've witnessed firsthand, actually, the advancements in LC-MS instrumentation and techniques. And collectively, I know we'd summarized these before, but, you'd mentioned gains in sensitivity, chromatographic resolution, mass resolution, speed of analysis, multiplexing and information density. The size of proteomics files are sort of gigabytes now. To set the scene, can you just share your thoughts on protein mass spectrometry and what that encompasses right now? And then, obviously we can focus down.
Yeah, as I think most people know, protein mass spectrometry really encompasses a very broad area, right? There's proteomics, which is a really important area. People are looking at thousands of peptides and proteins in a single experiment, and that's where I think you're getting at with the gigabyte data sizes. But there's also the characterization of biologics, right? So when you're doing bioprocess, right? So biologics, it's the process defines the product. So you have to really understand, how these different steps in the bioprocess, the downstream purification, how they're impacting the final product. And mass spectrometry plays a really important role in that, right? And then there's area of bioanalysis, which is what I focus on now, right? That's quantifying it or doing characterization within a biological matrix, like plasma, for example. So I think for today's discussion, I'd like to talk about primarily the bioanalysis of biotherapeutics and biomarkers in the way that we're doing it. And as you mentioned, over the last 20 years, there's been tremendous advancement in mass spec instrumentation, as well as the chromatography, right? We're using the UPLC, we're using low flow techniques, and the instrument manufacturers have really done a tremendous job in driving the performance of these instruments. So you have triple quads that we use very heavily, are much more sensitive than they were 20 years ago. The high resolution mass spectrometers are much more widely available. So you have QTOFs and Orbitraps that we have available, and these really help us kind of delve in and answer some key questions and drive assay sensitivity in a way that I think 20 years ago you didn't even think about.
Yeah, I started out in mass spectrometry probably a similar time to you, and it was a very different era. So the kind of current state of the art, the instrumentation and things that you mentioned there, they're obviously now heavily associated with the discovery and development of large molecules as therapeutics, something that's drastically changed over that period as well. So I wondered how you felt LC-MS has impacted the bioanalysis of those sort of biological modalities.
Yeah, I mean, biologics in itself is a really fascinating area, right? So over the last 20 or so years, there's been a dramatic increase in the investment and the amount of research done in the biotherapeutic area. Like, for example, in 2002, the FDA approved 7 biologics. Now, if you fast forward to 2022, that number is now 19 biologics. And I believe it was in 2022 that 50% of the FDA-approved drugs were biologics. So definitely a different landscape than 20 years ago when we were mainly talking about small molecules. Now, a lot of those biotherapeutics have become more complex in terms of the molecular structure, right? So you have things like bispecifics, multispecifics, and you're trying to bind multiple targets with a single molecule, right? You have ADCs, PROMABs, FC fusion proteins. Now, there's really no end in sight in terms of how creative you can be in terms of constructing these molecules. But then bioanalytically, how do you deal with these? And that's where I think mass spectrometry is definitely playing a role. So, for example, ADCs, what happens to an ADC once it's dosed, right? So about 10 years ago, Genentech is really kind of a leader in the ADC space, especially at that time. They published a really fascinating paper where they used intact mass spectrometry, so LCMS, looking at the entire molecule, and they were able to measure the drug to conjugate ratio, or the DAR, right? And what was really important about that work is that once the ADC was dosed, then the question is, how long do the conjugates stay on? Do they get cleaved off quickly? Are they going to withstand the whole duration of the dosing time?
Yeah.
So by using this intact mass approach, you're able to look directly at the molecule, and they were able to track the change in the DAR. And that's really important for ADCs because a lot of them have cytotoxic payloads, right? So you don't want the conjugate coming off while it's in systemic circulation. You want it to come off when it's in the target cell, right? That's the whole idea. So it allows you the ability to monitor it, and then maybe you can make some changes to optimize the molecule, or at least you're better understanding when you're dosing it what's happening to that molecule. Yeah. Now, ADCs were kind of just the start. I mean, there's all these other molecules I mentioned, right? So, you can use the same approach to try to answer questions related to some of these other complex biologics. So another one that I've worked on in my past job is on PROMABs, right? So these are really interesting molecules that they have peptides that are tethered to the antibody. And the goal of the peptide is to self-bind to the binding pockets of the antibody. So that inactivates the antibody. So it's kind of like a prodrug, a small molecule prodrug, right? Where you dose the phosphorylated form because the absorption through the gut is not that great. And then once it gets into circulation, it then gets activated. Well, similar concept with these PROMABs. But the thing is, you don't want it to cleave in general circulation. Like a lot of these are designed to go after solid tumors, right? So you want it to cleave when it's in the tumor microenvironment.
Yeah.
So how do you track all that? So this intact mass spectrometry method that Genentech pioneered, it really is well suited for this kind of question. So you can track, how it's coming off, where it's being cleaved. So it's a really powerful way that I don't think you can look at with any other technique.
Yeah, that's brilliant. And maybe chat a bit about LCMS approaches and which ones are applied to the bioanalysis of biotherapeutics, because I know there's a bit of jargon, that people tend to refer to: top down, bottom up, middle down. It might be useful just to kind of lay that out for our listeners.
Yeah, so mass spectrometry, at least for biotherapeutics, is applied in a variety of different ways, right? And you mentioned top down, right? So that's what we call the intact mass approach, right? So, and that's what was used primarily for ADC characterization. And then also allows you to look at clipping. You can also monitor the changes in glycosylation patterns on your molecule to see if that's being impacted. And then you can also do that in a quantitative manner, right? You can create a calibration curve of your drug, and then you can use that to back calculate the levels that are in your unknown samples. So it's a powerful technique to do what we call quant-qual, right? Quantitative and qualitative analysis in a single run, right? And then there's another area, another approach where you can digest your sample into peptides, right? And we call that bottom-up approach. So that's really useful for if you need to pinpoint the exact location of like a different modification. Like a good classic example is deamidation of asparagine or isomerization of aspartic acid, right? There's a tiny mass changes, but you can use this peptide-based approach or this bottom-up approach to focus in on those particular peptides and those particular sites. Another thing you can do with the peptide, and it's where it's, I think, widely used, is for quantitation, right? And the reason why people like to use the peptide approach is that it's much more sensitive than the intact mass approach, right? So then what you're doing in this case is you're focusing on a particular surrogate peptide, if you would, right? So you've digested the sample, and you now have a peptide that's selective for that biologic of interest. And then you use conventional standard curve, and you've converted this large molecule basically into a small molecule. So we typically use like a triple quad mass spectrometer for this kind of work, right? And if you combine that with immunocapture techniques, right, you can really push sensitivity. So it's not uncommon to get LOQs for biotherapeutics in the low nanogram per mil range. And there's one other key way that mass spectrometry used for biologics, and that is what we call like a middle down approach. So that's where we're looking at subunits, right? So typically what you would do is after you've immunocaptured your sample, you would then reduce it. So you'd have light chain and heavy chain. And what that allows you to do is it increases the ionization efficiency, because these are smaller molecules now. And you improve the resolution. So it allows you to localize modifications or clipping to certain regions of the molecule. And once again, that can also be done in a quantitative manner. Same thing, you generate calibration curves, and then you back calculate. So, there's so many different ways you can attack the question of what's happening to the biologic or what the concentration is of the biologic by using LC mass spec.
And does LCMS offer unique advantages for the bioanalysis of biotherapeutics, say in the preclinical space?
Yeah, I think mass spec's really well suited for the preclinical space. Especially for biologics that have a human FC, right? Because you can use an anti-human FC and readily pull out the biologic, right?
Yeah.
And, that allows you to develop assays very quickly. It really doesn't take that time at all. Another thing you can do, because of the specificity of mass spectrometry, is in the area of surrogate antibodies. So these are tool molecules, and typically it's like a mouse antibody that's going to be dosed into a mouse, right? But it's early enough on that you really don't have a lot of different reagents available, right? So, like for example, if you want to develop an LBA for this kind of molecule, typically you'd need some target protein, right? You do that for capture, and you'd use some kind of generic reagent for detection. But if you don't have access to the target protein, there's not that many options. But that's where I think mass spectrometry plays a role. So what you can do is you can actually fractionate like a subtype. You can use an anti-subtype specific antibody against like mouse. So let's say it's like IgG1A, mouse IgG1A subtype. You just pull that fraction out, you then digest it, and then you pick a peptide that's unique to just that antibody. And in doing that way, you can actually push the LOQs down to 50 to 100 nanograms per mil, and you can develop this very quickly. So I think, mass spec really is in position to help accelerate discovery programs. We were involved in a program where it was sort of this thing. It was a surrogate antibody, or actually a surrogate molecule. It wasn't necessarily an antibody, but the team approached us. They were telling us about this other strategy they had where they're going to put some kind of fluorescent label on the molecule, but that was going to take about 6 to 9 months to generate that molecule. And then we talked about this approach, and we were able to develop a method a week later. And then we actually knocked off like 6 months of time to conduct these experiments. So we had answers to some of these experiments much quicker. So I think if people know how to utilize these techniques, you can really benefit programs. Another area that is important is combination dosing, right? So, a lot of diseases, you have to tackle them with more than one molecule, like more biotherapeutic. But how do you do that? So mass spectrometry is a really convenient way because of specificity. You can pick a surrogate peptide from 2 different molecules and then set up a multiplex assay. A great example of that was Regeneron. They actually did it in a clinical assay. So they were developing their cocktail for COVID treatment. And how do you develop all these reagents in a fast period of time to do an LBA? Well, they fast tracked it, and they used mass spectrometry to do some of the initial development work in the clinic. So a really powerful example of taking advantage of the specificity of mass spectrometry.
Yeah. No, that's really interesting because the time advantages of reagent sort of free approaches, obviously, or less reliant on reagents, is like one of the big selling points, I think, of the technology. Biomarker quantification, that's another important area where LC-MS has been applied. What's the rationale behind the decision to choose LC-MS for certain biomarkers, do you think?
Well, as I mentioned, like selectivity, right? These are, or specificity of a mass spectrometer. That's really where I think you got to think about when you're looking to apply mass spectrometry, right? So a lot of these endogenous biomarkers, they could have post-translational modifications. They could be a mixture of isoforms, or they could be different truncated forms. Some of which only differ by a single amino acid residue. Mm-hmm. So it's hard to use reagents to kind of pick out these different forms, right? And mass spectrometry is really well suited when there's small mass differences between different proteins or between different peptides. So you can use that to your advantage, and you can develop an assay for a specific isoform, so that may be the more active form of the biomarker that you're interested in. Another advantage is in peptide analysis, right? So these endogenous peptides, these bioactive peptides, if you would, if you want to develop an LBA, ideally you want to have 2 different reagents, right? A capture and detection. But these are small enough molecules that you don't always have the luxury of independent epitopes on such small. Now you can use what's called a competitive ELISA, right? You can develop one of those. That's where you use a single reagent. But there's always caveats when it comes to the selectivity of those kinds of assays. Where mass spectrometry, alternatively, you can get by with using one capture reagent, and then you use the specificity of mass spec for detection. So you can readily develop these kinds of assays. And then you don't necessarily even have to have a capture reagent, depending on what the sensitivity requirements are for the assay, right? You can do some kind of basic cleanup, like you can do a protein precipitation. You can do a solid phase extraction as a simple cleanup. And then once again, use the specificity of mass spectrometry. So in that case, you typically can, achieve like 100 nanograms per mil detection limits or quantitation limits for endogenous peptides. And then multiplex is another area, right? That's an important area. So, it's not a stretch to think about, analyzing or quantifying like 10 different species in a single run, right? And there's some really interesting work that's coming out of the Fred Hutch Cancer Research Center with this woman that runs a group called Amanda Polovich. Her lab, they've been working on developing assays that have tens of, I think even up to 60 biomarkers in a single multiplex assay. So this is, well within the capabilities of mass spectrometry to develop these kind of multiplex assays.
And one of the other big changes we've seen in the time that we've been in the industry is the kind of idea of the 3 pillars of drug survival, 5 Rs, sort of pharmacology. And it's becoming common now for companies not to advance molecules into the clinic without target engagement biomarkers. And biomarkers are also used for patient stratification as well. So as a result, there's increased demand for developing biomarker assays. What's the impact of mass spectrometry in the biomarker space?
Well, as you mentioned, there's a greater role of using biomarkers to progress these drugs into development space, right? But also in the discovery space, biomarkers can be important. Like, for example, if you're trying to establish the proof of concept, right, of a certain target mechanism, right? It's nice to have a biomarker that indicates that you're impacting that biological pathway, right? And it's going to help you when you try to make the decisions whether you're going to progress an asset or whether you're going to deprioritize it, or maybe you need to do more fundamental research to understand that mode of action, or to understand if you have the correct or the best animal model, right? So biomarkers definitely play a role in there. Biomarkers, there's a whole breadth of different technologies that you can use to measure biomarkers, right? So there's, like, for example, cell surface biomarkers. You use flow cytometry. For RNA, using genomic approaches. There's histology, LBA. So you have all these different platforms, but mass spectrometry is still an important part of that arsenal, right? And that's really going to be important when there's a lack of suitable reagents, or that specificity is really a key question to this. So that's where mass spectrometry plays a role in the biomarker space, along with these other platforms. I mean, it excels really well when there's small mass differences between different proteins or peptide biomarkers, right? And even if you have the same mass, so-called isobars, right? 2 species with the exact same mass, you can still use the chromatography to separate these 2 different species, and then use mass spectrometry for detection, right? So another example of using mass spectrometry for biomarker space, like for target engagement, is when you have, let's say you have an enzyme that cleaves a particular protein biomarker. So what you can do with mass spec is that you can develop an assay for the truncated form or the cleaved form, and then an assay or a measurement for the full length, right? And you can do that in a multiplex manner. So you're going to measure both the intact and clipped forms, and you're going to see how, like, for example, if you're trying to inhibit that enzyme, right? The enzyme's your target drug, or the target of your drug. You can see firsthand how that biomarker demonstrates that you're engaging that target.
Yeah, it's a really nice example of using the mass domain, for measurements of, at that level of selectivity. Sort of exquisite selectivity. You and I were mass spectrometrists, and that's obviously been something we've grown up with. But to be fair, in the biological space, it's not always the most sensitive platform. So I was just wondering how can sensitive LC-MS methods be developed, and the kind of practical limits that you can achieve.
Yeah, sensitivity can sometimes be showstopper, right? You have great specificity, but then you just don't have the low levels that some of these biomarkers are present, right? So there's really three main ways that you go about trying to improve sensitivity. First of which is immunocapture. So if you have a reagent that you can selectively pull out the protein of interest, that's going to provide a big improvement. There's multidimensional separation and low flow chromatography, right? So immunocapture, that's going to allow you to remove all this background material, right? So if you're working with plasma, for example, because we all know that you have albumin, but you have these other proteins, you have lipids, salts, carbohydrates. So you can remove all that. And the key there is that, you're not going to overload your LC mass spec system with all this extraneous material, right? You want to just hone in on loading the sample that just contains or primarily contains your biomarker protein. Right? Another thing it allows you to do is use a much larger volume starting sample, right? So there's many clinical examples where you're using something like 500 microliters of plasma or serum. And then what you're able to do is by using immunocapture is you can concentrate it down and then elute it off in like 50 microliters. So now you've got like a 10-fold bump in concentration. And most of that you can load directly on the mass spectrometer. The other way that we use is multi-dimensional separation. So that can take a variety of forms. That can be 2 different levels of reverse phase. So you can run reverse phase at 1 pH, like a higher pH, and then run a second stage of reverse phase at low pH, right? You can do this offline. You can do it online. You can combine ion exchange with reverse phase. These are much more orthogonal separation modes. You can combine solid phase extraction, this part of this process. So there's a variety of different ways to do it. There's a technique called prism, which we try to use. That's where you use you generate or you synthesize the heavy labeled form of the peptide that you're trying to monitor, right? You've digested your protein, right? You're now working on a protein peptide level. So you're trying to fractionate. And then the question is, what fraction has your peptide of interest? So if you spike in these heavy labeled peptides, you can use that as a tracking probe. You can pinpoint where the fractions of interest are. And then you take those fractions, right? So you've done, like I mentioned, a high pH fractionation. You now know what fractions contain the peptide. You then take those and then you can inject them, usually on a lower flow system, like a microflow system. So, I mean, it's a lot more work than doing immunocapture. Immunocapture is actually, you're allowing the antibody to do the bulk of the work. But in the absence of reagents, you do have these other options. And then the low flow techniques are really a game changer, right? So, you do this sample cleanup and then you inject the samples on these low flows. So you're talking about 1 to 5 microliters a minute. Typically you can get something in the order of 5 to 10-fold improvements in signal to noise if you do this properly, especially if you marry it with immunocapture or, significant cleanup with these multidimensional techniques, separation techniques. But as far as like how low you can go with sensitivity, you can develop some pretty exquisitely sensitive assays, but it's going to take experience. It's going to take know-how. It's going to take using the right kind of platform. And there's a group out of Pfizer that's done a tremendous amount of work. It's a group with Henrik Newberg. The focus of their platform is they rely on these anti-peptide immunocolumns, these immunoaffinity columns. And the way they have that set up is, these columns, they're standard bore columns. They're like 2 millimeter diameter columns. So you can load these at higher flow rates, like 500 microliters a minute. And then with some column switching, you can then direct a fraction of that onto a nano column. And then you can really boost the sensitivity. So you've cleaned up the sample extensively, and now you're using the most sensitive form of chromatography combined with mass spec. And what they've shown is they can push sensitivity levels down to like 0.5 picograms per mil. Okay. Now, if in our lab we're not using that technique, we're typically using microflow. So for us, we typically are going to achieve LOQs from 10 to 100 picograms per mil. Yeah. If we've done our sample prep properly. Yeah. So, it's not uncommon to be able to push sensitivity. It's going to require more work and resources, but it can be achieved.
Yeah, I mean, in molar concentrations, that's astonishing levels of sensitivity.
It would be nice if some of the vendors would kind of commercialize that approach, because this is really kind of they're taking off the shelf components, right? They're stitching them together, but it does take know-how and resources. But once they've got it set up, then I think it's like anything, maintaining and knowing how to use it is much more doable. So if one of the manufacturers wanted to try to develop this and market it, I think there'd be some buy-in and would be some real benefit.
John, there's a real call to arms. Let's hope some manufacturers listen to the podcast. I was thinking about the future. We've talked a lot about this kind of state of the art, and that's a really useful summary for our listeners on where we are and what you can do and what you can achieve. So turning an eye to the future, where do you see the growth in applications of mass spectrometry for biomarkers and biotherapeutics?
Well, I still see some expansion in the use of these microflow and low flow techniques, because there's been some great published examples of using them. We just talked about sensitivity, so that's the big driver. But one of the things you have to keep in mind is that you really have to have very clean samples in order to take full advantage of the low flow techniques. And I think the manufacturers, so about 10 years ago, there was a push by manufacturers to sell these microflow systems. So they made them much more rugged and easier to use, which was great. And I think they did a great job in that aspect of it. But I think some of the marketing behind it at the time was they tried to push it as something very broadly applicable. So you could take a sample that you're running at high flow rate. Let's say you did an SPE cleanup step. The marketing was that, you can just take that and put that on a microflow system and you're going to get a boost in sensitivity. Well, you do typically see an increase in the intensity, but at the same time, you also boost up your background signal if it's not real clean. So there's no real net gain in signal to noise. So I think some analysts got a little discouraged with this because they tried to apply it in a way I think was not optimal. But I think now it's realized that you can gain the sensitivity and push LOQs, but you just have to do it properly. So I think with that realization, you will see a continued increase in the microflow techniques. As far as the biologics go and the biotherapeutics, you typically don't need that level of sensitivity. So the microflow, I don't think is going to be applied too commonly for biologics. Mainly because, the dosing levels we're talking about, you're at least 1 mg per kg, a lot of times 5 to 10 mg per kg. And, the way we're doing it now is more than adequate to achieve the LOQs necessary. So you don't have to employ the microflow. I talked earlier about the top-down approach, right? The intact mass. That's still going to play an important role with these complex molecular structures that the protein engineers are developing. Right. So that it's always going to help you have greater understanding of your molecule, especially once it's been dosed. And I also mentioned the combination dosing. I think mass spec's really well positioned to help drive that, right? Because, I see a lot in the cancer therapy, immuno-oncology, that you're using multiple agents. If you want to speed that up and get to the assays quicker, I think mass spectrometry is more than well positioned to do that. And then I also talked about the multiplex for biomarker analysis. And I talked about the group out of Fred Hutch's Cancer Research Center. So, they're realizing that individual biomarker readouts are not necessarily informative enough. So people are starting to look more at panels of biomarkers. And the idea there is you can look at, maybe up to 100 proteins, right? And what the Fred Hutch group is doing is they're looking not only at the protein, but they'll also look at like the phosphorylated forms of those proteins, right? And what they're doing is they're looking at there's different biological pathways, and they're applying it to cancer treatment. So they're monitoring these patients as they undergo the treatment. And the idea is that, how do different individuals respond to this? Because everyone's going to have their unique response as a patient. But really, they want to take these learnings to help drive precision medicine. Now, there's also all these non-mass spec proteomic platforms like Olink, proteomics is another example, right? They use this DNA, these tethered DNA pairs that bind, and then they can amplify them using PCR methods. So it's a really fascinating approach. They make these protein panels. They can really push sensitivity quite low. Other examples are a SomaScan. They're using aptamers as their capture and detection reagents. So they're able to look at hundreds of proteins in single experiments. And that's really important for trying to understand like the molecular origin of diseases, to understand how individuals respond. But I think as we know that sometimes just identifying and measuring the protein is not necessarily the whole picture, right? Sometimes it's the post-translational modifications. And that's, once again, I think where mass spec plays a key role. So I think it's going to be the combination of these different omics platforms along with mass spectrometry, looking at, specific questions about particular proteins. I think combined, that's going to help enable precision medicine.
Yeah. So while we're talking about the future and we've got you, I thought it'd be useful to hear your views on exosomes and extracellular vehicles, because it's quite a new area, but it's one that mass spectrometry may be able to contribute to as a source of biomarkers. So what role do you think they'll play in that space?
Yeah, I think exosomes and these extracellular vehicles, or vesicles rather, these EVs, they're significant because they're considered a potential source of biomarkers, right? And they're thought that you can use these as liquid biopsy, right? So that's important when you're talking about the need for tissue biopsies, right? Like, for example, in liver disease, it's pretty common that patients have to undergo a liver biopsy, and they may have to do multiple liver biopsies, maybe early during diagnosis and then later on during treatment, right? But it'd be nice if you could reduce the need to do some of these tissue biopsies. You may not fully eliminate it, but if you can use a liquid sample, like plasma, for example, that would be a real benefit to the science. It would be a real benefit for patients, right? Because the thinking is these exosomes or these EVs are these capsules that are excreted by cells, but they carry cargo. So the protein, there's RNA, DNA in these capsules. And in circulation, they're going to provide this protective shield around the cargo that's in these exosomes. But it's unknown what proteins are in there. And they're thought to represent the tissue of origin. So like I mentioned, liver disease. So if you have exosomes that are coming from liver, they're thought to be these tiny little liver particles, so to speak. So, there are going to be some unique markers that are attributed to the source, right, to the liver, but then there's other cargo in there. So I think that's where mass spectrometry is going to help to characterize the protein content of these exosomes, but also then you can use them for quantification, in concert with other techniques, other measurements. But clearly, to understand the proteomics and the protein composition of exosomes, mass spectrometry is going to play a key role.
John, thank you so much for sharing your experiences and also insights on such an important area of analysis. Thank you, John.
Oh, it's been an absolute pleasure, and I really appreciate the invitation to participate in this podcast. Thanks so much.
No, you're welcome. You're very welcome. And to our listeners, thank you very much for taking the time to hear John. Just a reminder that our podcast series is available on demand, answering a variety of key DMPK related questions important to drug discovery and drug development. So stay tuned for more topics in PharmaRun's DMPK Insight series, and bye for now.